Hello, my name is Yang Wang. I'm a second year medical student at the University of Pennsylvania Perelman School of Medicine. I'd like to give you a brief presentation on the application of liquid biopsy to the clinical management of brain tumors. First of all, what is liquid biopsy? Broadly speaking, liquid biopsy means sampling tumor-derived materials from body fluids, such as blood, urine, or cerebrospinal fluid, to make inferences about the pathology of an underlying malignancy. The research on liquid biopsy was driven by the early discovery of tumor materials in peripheral circulation. As early as 1977, researchers had discovered a link between the concentration of serum cell-free DNA with metastasis and response to therapy in several cancer types. In 1994, it was shown that KRAS mutations are detectable from the serum cell-free DNA of pancreatic cancer patients. Of note, that the great majority of cell-free DNA are not of tumor origin, but come from normal, healthy tissue. This means that detecting circulating tumor DNA from the pool of all cell-free DNA is challenging, almost like finding a needle in a haystack. Fast forward to the present day, we're now able to study a bigger panel of liquid biopsy biomarkers thanks to improvements in detection technologies. This includes circulating tumor DNA or RNA, which, as we mentioned a moment ago, is a subset of all peripheral cell-free DNA. There are also circulating tumor cells or CTCs, extracellular vesicles such as exosomes, and tumor proteins and metabolites. There are currently 839 registered clinical trials on clinicaltrials.gov that leverage CTC or ctDNA in one way or another. In this presentation, we'll focus on cell-free DNA, circulating tumor cells, and extracellular vesicles, and discuss how they could contribute to the clinical management of brain tumors. Liquid biopsy has several advantages over traditional biopsy. It is minimally invasive, requiring only the collection of bodily fluids to obtain the necessary sample, whether it's through blood draw, urine collection, or a spinal tap. This renders liquid biopsy an appealing alternative to traditional stereotactic brain biopsy, which could be too invasive for certain patients to tolerate. In addition, when tumor materials are detectable, liquid biopsy can theoretically better characterize tumor heterogeneity than tissue biopsy, which can suffer from tissue sampling bias. This is important because glioblastoma, the most common primary malignancy of the brain, is known to harbor very high intratumoral molecular heterogeneity. Liquid biopsy, by definition, samples materials released from the entire tumor and thus is expected to represent the tumor's mutational landscape more truthfully. Finally, unlike using traditional serum markers such as CA19-9 for pancreatic cancer and CA125 for ovarian cancer, the ideal liquid biopsy biomarker is derived directly from the tumor and hence tend to be very specific. It allows clinicians to apply well-established analytic techniques such as sequencing and PCR to directly investigate the mutation status and mutation load of a tumor. This very important information can then be used to guide therapy or inform prognosis. A major challenge of using liquid biopsy for brain tumors specifically is that the blood-brain barrier limits tumor material extravasation into peripheral circulation. One study from 2014 claimed that circulating tumor DNA is only detectable in less than 10% of glioma patients. This would imply that there is very little tumor material available in peripheral circulation for biopsy to begin with, and the biopsy will not be very sensitive. While sampling from the CSF can potentially improve tumor DNA yield and detection sensitivity, it is impractical to do liquid biopsy with periodic lumbar punctures or drains. On a more positive note, more recent studies utilizing improved methods of detection have shown better detection rates. GBM-related mutations in key driver or resistance genes such as TP53, NF1, EGFR, BRAF, and IDH1 have also been identified in plasma cfDNA. Another study that leverages digital PCR was able to detect TER mutations in the plasma cfDNA of glioma patients with 62.5% sensitivity and 90% specificity, showing that it's indeed feasible to detect circulating brain tumor DNA with clinically relevant sensitivity and specificity. MGMT promoter methylation status, an important predictor of GBM's response to temozolomide treatment, can also be detected from the serum, showcasing that tumor cell-free DNA can also shed light into the GBM epigenome. Even without explicitly detecting tumor-derived cell-free DNA, just the bulk plasma cell-free DNA by itself can also provide prognostic value. Studies have shown that plasma cfDNA concentration is associated with response to therapy and radiographic GBM progression, and preoperative baseline cfDNA concentration is associated with both overall and progression-free survival. The correlation of cfDNA level with progression is especially important as it could help distinguish true disease progression from pseudoprogression, which is a radiographic finding reflective of GBM's response to therapy. If tumor DNA are so rare, where does this increased cell-free DNA come from? Interestingly, there has been evidence showing that DNA may come from peripheral neutrophils undergoing mitosis, a pro-tumor immune response that could be implicated in GBM tumorigenesis. In addition to circulating tumor DNA, circulating tumor cells are also widely studied. Can we find CTCs derived from brain tumors? Studies to date have shown positive results in detecting CTCs derived from metastasis of epithelial cancers to the CNS. This is in part due to the availability of epithelial surface markers such as EpCAM and keratin as a part of the cell search system, which is the only FDA-approved CTC detection platform to date. In general, studies have shown that CTCs from epithelial leptomeningeal metastasis can be reliably detected from the CSF with good specificity and sensitivity. On the other hand, GBM as a mesenchymal malignancy does not have definitive markers, which makes GBM CTCs difficult to capture and enrich. A pilot study in 2021 showed the use of GFAP and surface vimentin as promising markers, but more research is needed to verify generalizability. To date, GBM CTCs have not been definitively associated with any diagnostic or prognostic clinical correlates, but studying them could still provide invaluable insights into GBM biology. Lastly, extracellular vesicles are another important set of liquid biopsy biomarkers that are frequently studied, with the exosomes being perhaps the most studied EVs. Exosomes are membrane-bound vesicles made by healthy and malignant cells alike that contain various DNA, RNA, and protein cargoes. They've been shown to play an important functional role in maintaining the tumor microenvironment across all phases of cancer development. It's worth noting that just by the nature of DNA extraction techniques used to isolate cell-free DNA, DNA molecules that are within exosomes are often extracted and studied as a part of cell-free DNA. Nevertheless, exosomes are prognostic in their own rights and have much more to offer than just DNA. Some peripheral EVs have been shown to be of GBM origin, and their quantities is correlated with GBM progression. Exosomal contents also carry clinical significance. These include microRNA species such as microRNA 21 and microRNA 29b, both of which are shown to be prognostic. Variant EGFR RNA transcripts can also be detected in CSF and serum exosomes from variant-positive glioblastoma with good specificity and sensitivity. Last but not least, GBM exosomal surface PD-L1 has been shown capable of binding T-cell PD-1 and is hypothesized to play a role in GBM immune evasion through PD-1 blockade. Exosomes have much more to offer, and we're only scratching the surface here. In summary, in this presentation, we discussed liquid biopsy as a minimally invasive method to sample brain tumor materials from body fluids, providing clinical utilities such as early tumor detection, mutation profiling, and progression and therapy response monitoring. Both bulk and tumor-derived cell-free DNA, as well as exosomes, are shown to be promising diagnostic or prognostic biomarkers. Circulating tumor cells of primary brain tumors, on the other hand, are difficult to detect in peripheral circulation, and we're still investigating their clinical significance. Thank you for listening, and I hope this presentation was interesting to you.